All right. So, couple things. We had a quiz, right? First quiz was due, I guess, yesterday. If you haven't done it, go ahead and do it. You can take the late credit for it. I have a second quiz that's posted that covers the introduction to the platyhelminthes. 10 questions. I made that due Friday at the end of the day, so 11:30 at night due date. So kind of reviewed platyhelminths. And with that, I want to just kind of mention something here. So we went over this, the systematics, and pointed out how all of our parasitic groups kind of form a monophyletic group. It's termed the Neodermata. And then kind of went through and talked about some of the key characteristics. That is out. So don't bother trying to memorize all these key characteristics. I'm not going to ask about them. It comes up as part of our actual species. So the adults are polyzoic. You'll know that from the cestodes and so forth. But at this point, I'm not going to ask any of these questions on here. So took that out. All right. All right. So we left off here. Aspidobothreans. Right? Where did we leave off? Did we do this? We did the body forms, I know. Right? Did we talk about the life cycle, generalized life cycle? So we picked up here. All right. So the Aspidobothreans, most of them have a direct life cycle. So we only know about 80 species or so. Most of these have direct life cycle, meaning what? How many hosts are in that life cycle? One. All right. There are a couple that have a multi-host life cycle, and one of them that we'll go over today. Regardless of if you have one or two hosts in the life cycle, we have the same general pattern in terms of the progress of our larval stages, the procession of our larval stages. So the egg comes out from the adult, all right? And then the egg is going to hatch and release a cotylocydium. And then that cotylocydium will develop into the adult. Now, it may stop. It's kind of like a pre-adult, kind of not fully sexually mature, but it's egg, cotylocydium, and an adult. So in those one-host life cycles, a lot of times what's going to happen is the egg hatches, and the cotylocydium is going to swim around, find that host, and then penetrate it. All right? Or get caught up in, let's say, respiratory chamber if we're dealing with an aquatic mollusk. Other times, the egg is going to be consumed. The cotylocydium is still there. It's going to hatch out of that egg, and then the cotylocydium is typically going to burrow into the proper location of that host and start to develop into our adult stage. Now, what is this cotylocydium? As you're going to see, there are several different names for this larval stage after the egg, and that name depends on what group we're talking about. So in our digenes, it's a myricydium. For our monogenes, it's an oncomyricydium. For our cestodes, it's going to be a coracidium or a hexacanth larva. All right, so there's several different names. In our Aspidobothreans, it's called a cotylocydium. What distinguishes this from all of the rest? Most of these things have ciliary tufts. That's what these are. So it's not, you don't have cilia all around it. The cilia are restricted to these very localized regions, making it look like they have basically like a ponytail. Right? These are ciliary tufts. These larval stages, unlike some other larval stages, have a mouth. They have a pharynx. They have a simple gut, a blind gut. All right? And they also have this prominent posterior disc, like a sucker. All right, now this disc doesn't have any of those alveoli in it. It doesn't have the loculi. It looks like more like a sucker, like our digenetic trematodes. The Bayer's disc is going to form from this structure. And what will happen is it starts getting those loculi form, the septiform, or maybe the rugiform, and then it kind of spreads through the rest of that worm. It's notable that this one does not have hooks, and that's unlike our cestodes. Our cestode larvae actually have hooks used for penetration. So any penetration that this guy's going to do, they don't accomplish it through the use of hooks. More than likely, it's going to be some sort of chemical penetration, releasing enzymes that dissolve some host tissue so that it can burrow through. There's no asexual reproduction in this life cycle. No asexual reproduction. So it is the adult, they release the eggs, that cotylocydium then develops directly into our adult. It's metamorphosed into that adult. Questions? Generalized life cycle, that's our larval stage. Example, Lobatostoma mantari. This is an Aspidobothrean found down on the Great Barrier Reef. I think I'm gonna try over here. It's found on the islands around the Great Barrier Reef. This species is pretty darn host specific. Right? It's only going to infect one definitive host, one type of definitive host, and that host is our snub-nosed dartfish. So our adult worm is going to be in the snub-nosed dart found in the small intestine. Great place for our parasites because when it releases the eggs just pass out with the feces. Right. The eggs are going to be out in the environment, and they're going to sit there and just basically wait until they get consumed by our first intermediate host, which is a marine snail. Work in the marine snail. Now what's going to happen inside this snail is that egg will hatch in the small intestine. All right. See if the red one will work. Oh, yeah, that's better. So our egg is going to hatch in the small intestine and release the cotylocydium, which then burrows out into the snail's digestive tissue, the hepatopancreas, and develop into a pre-adult. All right, so inside this snail, we'll have our pre-adult in the hepatopancreas. All right, so our hepatopancreas it's a snail's digestive tissue. So in a snail, you've got the body basically in the shell, coiled in the shell. All right, you've got the gut, and then around the gut is this hepatopancreas, the liver, pancreas. It's our digestive tissue. That's where nutrients are absorbed and everything. And then as we go posterior to that, then we have our gonads developing. These guys are in the digestive tissue, which is the bulk of the snail itself. All right. Now, what is this pre-adult? The pre-adult is basically looks like our regular adult, smaller. And it's not mature. So its reproductive organs, they've developed, you can see them, but we don't have our eggs yet. I guess maybe not mature is not going to be the best. Reproductive. not reproductive. So we have our testes and ovary, but no eggs. And that's kind of interesting because, as I said, most of these Aspidobothreans have a direct life cycle. All right, so you go from one host to the next, and you develop right to the adult. How did that develop? It's likely that it developed a one-host life cycle first, and then you start to add a second host. What if we're in the middle of this life cycle, adding a second host, where our adult can develop, but it doesn't develop fully. It doesn't reproduce, so it just stops. And then eventually, it'll get to the step, let's say thousands of years from now, where we don't even get to this stage, but it develops to, let's say, sexually immature, no ovaries, no testes, more like the genital primordia. And then once they get back into here, then we develop into the adult. So we're at the pre-adult stage, gets to our snub-nosed dart when it consumes this snail. Now, I said, this is an extremely host-specific life cycle. Why is that? This fish has pharyngeal plates. So inside, basically, its throat, it has these very hard plates that it uses to crack the shells of the snail. So this fish itself is a specialist on the snails. Other fish, who knows? Maybe they could survive. Maybe they can consume or get infected. At least in the experiments, I don't think they've been successful. But this guy specializes on these snails. And is one of the few that specializes on the snails, and it's the only one that gets infected. All right. Questions on our life cycle. The definitive host is our snub-nosed dart. Eggs get passed in the feces. Eggs get consumed by our snails. We have our life cycle up to the pre-adult stage. Once we get to the pre-adult stage, we are now infective. And when a snub-nosed dart comes along and consumes one of these snails, it can be infected. All right. Questions? So, Lobastoma mantrai on the Great Barrier Reef. Any questions? All right. No questions? All right, so that wraps up the Aspidobothreans. All right, so we had, the class Trematoda, subclass Aspidobothreans. The other subclass is Digenea. Oh, so that reminds me. We finished up the Aspidobothreans. That quiz goes live at noon today. You have until the end of the day on Monday. So kind of you can cover up for that second quiz this week, and then when you finish that one, start reviewing the Aspidobothreans. All right, so now we're going to move to the subclass Digenea. Why is it called Digenea? 2 generations. That's what it's named for. And as a preview, that's suggesting we're going to have a generation of sexual reproduction and a generation of asexual reproduction. That's going to be the rule in our digenetic trematodes. All right, so most of these worms are endoparasites, usually in the gastrointestinal tract, so the GI tract. Could be mouth, could be esophagus, could be stomach, could be small intestine, large intestine, and so forth. So just in the GI tract. Most of the time, these are in the small and large intestine. Small intestine. Vertebrates are going to be our typical definitive host. Many types of vertebrates are going to serve as hosts. Our adults usually have 2 suckers. You're going to have an oral sucker where you'll find the mouth, and then you'll have some sort of ventral sucker, also called the acetabulum. All right, so in this diagram, this is Helicometra. You got your oral sucker, the mouth is located up there, and then you have our ventral sucker, or the acetabulum. That ventral sucker could also take on the name of opisthapter. Kind of confusing, but for this class, it'll be ventral sucker acetabulum for our digenetic trematodes. Our opisthapter is a monogene attachment organ. So then in our Aspidobothreans, we'll talk about Baker's disc or ventral suckers and those to kind of start helping you learn differences between these groups. Now in our digenes, size and shape is highly variable. Depends on our species. All right. Some species, the adult is only a few millimeters, if that. Others, several centimeters in size. So we already saw one that's pretty large. What species was the large one that we looked at in the lab? What's that? I heard someone say it. Yes, I can't get a single picture. Fasciola, giant liver fluke. So several centimeters in size. Now that's big, but we also have some other worms that are several centimeters that are really thin. All right. They could also be round in shape. They could be leaf-like in shape. They could also be ribbon-like in shape. We've kind of started seeing some of the variation in our patterns in the shape. And just to kind of throw you a curveball on here, it seems to have some morphological variation depending on what host you're in too. So the adult worm develops one size, one shape in one host, and it will be slightly different in another host. How many are those? It's a good question. A lot of times if you find a worm and it looks different than what you found in other hosts, it gets classified as a new species. But with genetic analysis, they're starting to see that some of these are actually the exact same species. You've got some sort of environmental effect leading to those changes. So I bet as more and more of these adults, as people go back and review some of these adult worms and start sequencing, maybe we'll find a lot more of this variation. Continue. And again, if I flip too fast and you didn't get it, please stop me. All right, so most of our worms are monoecious. All right, we've already said these. This will be the case where our worm contains both male and female reproductive organs, so they're hermaphroditic. They can self-fertilize, although cross-fertilization is probably preferred. All right? And in all of our digenes, we have a complex indirect life cycle. In many cases, it'll just say a complex life cycle. That implies it's indirect. All right? But all of our life cycles require at least two hosts. And one of those hosts, that first intermediate host, almost all of them require a mollusk. I didn't say all of them because I'm kind of protecting myself against that one or two that I don't know about. But it seems like all the ones that I know about, they always go into a mollusk as a first intermediate host, so either a snail or some sort of bivalve. There's going to be anywhere from 2 to 4 hosts in the life cycle, plus possibility of paratenic hosts. This is a live worm pulled out. You can see, we've seen the slides that have been cleared and stained. A live one's going to look something like that usually, whitish, pinkish. You can kind of make out the eggs. This is the uterus. This brownish to get to the blackish stuff. You can make out the sucker. Sometimes you can make out some other structures in there as well. All right, so the holdfast. These guys use suckers to attach and remain where they're at. Usually, our worms have 2 suckers, an oral sucker and then a ventrally located acetabulum. Now, that location, the position of that acetabulum is going to vary among species, and that's led to a couple different names. So just in a generic terminology of the shape, we can have a diastome, 2 mouths, because they thought you had mouths in both of they thought these suckers were actually mouths, but they're suckers. All right, so there's a diastome. You've got your oral sucker, and then you have a mid-ventrally located sucker. So someplace in the middle of the body, could be in the upper third, could be right in the middle, right? Or we can have an ampistome, where we still have our two suckers, but now our ventral sucker is located at the posterior end. Almost makes you think it looks like a leech, but it's not. Right? It's not. We lack our segmentation. And then you have some where you only have one sucker, an oral sucker, and that's called a monostome. And again, if we do systematics on it, it's not the situation where all of our monostomes form a monophyletic group. It's suggested that this loss of the ventral sucker actually represents a secondary characteristic, some sort of derived trait, and it's happened more than one time over the course of evolution of this group. So those are your hold fast, and in addition to the suckers, many of our adults also have spines on their tegument. And it's thought that these spines assist the worms in helping to lodge in the intestine. So if the spines are all pointed backwards, you can kind of get into the villi, the microvilli of the intestine, and hang out there, and those spines help to kind of hold you in place. They hook on those villi and prevent you from drifting back with the food that's being pushed peristaltically. Right, so our hold fast suckers could be supplemented with some spines on the body. Tegument of these digenes is a syncytial tegument. All right, we've already talked about it. This is a diagram from another textbook, but you can have your anucleate layer, your spines, your muscle layer, your cytons with the nuclei, and so forth. All right, and what we're going to point out is this glycocalyx. So the glycocalyx is the living plasma membrane that's interspersed with protein-rich elements. It's going to be this layer right at the surface that's going to be constantly shed over the life of our adult worm. All right. Now, it could be shed because it's been damaged from enzymes and stuff, so it's just replacing itself. All right. And that's probably the response. But the added benefit is as we have our host immune system responding, antibodies binding, effector cells binding, they get lost with that glycocalyx as it gets shed and goes out with the rest of the food waste. All right, so if you need to review what the tegument is, go ahead, check back the introduction to the platyhelminths, where we describe it and talk about the functions. Glycocalyx layer is going to be important for this group, as it is for our cestids. Digestive system.
Can you go back one? I'm sorry.
Yep.
And that's like a form of protection, correct?
Yeah, the glycocalyx.
Mhm.
Yep. The function. That's why it says it might have evolved as protection, like it was damaged, and that's why it's constantly sloughing off. And then that added benefit is that it's taking the host immune cells with it. So yeah, it's part of the protection. I like this drawing because you can also, so the mitochondria are marked as M right here. And you can see that the mitochondria also extend into that anucleate layer. So it's not just restricted down into those cytons. It says it's producing stuff here. It needs the energy up in this area. All right, we good? All right, digestive system, this guy. Consists of a mouth, a prepharynx, a pharynx, and esophagus, and a ceca. Now, how long or even if the prepharynx and esophagus is there is species specific. All right, it's going to be species specific. So, for example, fasciola, you don't have the esophagus. Now, what is that? Well, you've got your mouth up at the top, located inside the oral sucker. That space between the mouth and the pharynx itself, that tube represents our prepharynx. I guess for those of you who are watching, here it is. So that would represent our prepharynx. Our pharynx is a muscular structure. All right, it's going to act by sucking food in to the mouth. So the mouth attaches, and then consider it like a suction cup to try to suck food in. If it's feeding on, let's say, epithelial cells, all right, it's going to generate a lot of suction to break those epithelial cells away from the host, and which incidentally can cause damage to that host. All right, so our prepharynx is a tube between the mouth and the pharynx, and then the tube between the pharynx and that fork is our esophagus. All right, and then you have your intestinal ceca that run down the length. All right. Fasciola doesn't have an esophagus. Its pharynx is right at our cecal junction, right here. So it goes from the pharynx and then immediately it goes off into our ceca. Sometimes your pharynx is going to be immediately at the mouth. That means prepharynx isn't present. All right, so you get nutrients through the consumption of food, but that's not the only way in which these worms acquire nutrients. They can also absorb nutrient through their tegument. Yep.
You say just prepharynx present, the variation species?
Yeah, both the prepharynx and the esophagus. So it's where this pharynx is and where the fork is in our species. So if the pharynx, like in fasciola, is right here at that fork, at that junction, there's no esophagus there. Others, our pharynx is located right at the mouth, so you don't have a prepharynx. But we present the digestive system to show that our worms can ingest food. It can also absorb nutrients through the tegument. Right? Absorption of food is also one of those functions of our syncytial tegument. The excretory osmoregulatory system is actually a combination of processes. All right, so diffusion through the surface to the outside. That's one way in which the organism can excrete some of their waste, their byproducts, byproducts of metabolism. They could also allow diffusion into the lumen of the intestinal ceca. Now, if it does that, how do you get it out? You get it out of the parenchyma, but how do you get it out of the worm? I'm going to go back to one slide here. So we've diffused waste into our intestinal ceca. How do we get it out? Take a guess. What's that?
The excretory bladder?
How do you get it from the ceca to the excretory bladder? So the excretory bladder is going to be connected to our flame cell system. So our flame cells, like the third part of it, it's going to collect, and that's what all of these are. All of these lines are part of our collecting system. I believe that's where they are. Good question. What these are? Yeah, excretory system. Yeah, so typically, so they drew it. I guess they don't have the connection here, but our collecting tubules connect down to the excretory bladder. So the flame cell system goes to this bladder, and they do have a couple points because there are some that have two pores, a primary and a secondary excretory pore. This might be one of those. But if you diffuse the waste into the ceca, how do you get it out? What's that? Through their mouth, exactly. They not only eat through their mouth, they also poop through their mouth, if you think about it. Pretty gross. They're not the only organisms that do this. They're not the only organisms that do this. But in this group, that's what has to happen. Especially if they can't diffuse it through their tegument. All right, so our excretions primarily through these processes. The flame cell system is primarily osmoregulatory in nature. It's going to be removing excess water. The ammonia waste, or the nitrogenous waste product, is ammonia in these guys. And they could do that. Ammonia is pretty toxic, but they're living in a place where that ammonia gets swept away from them, gets taken out. Any questions before we get to the reproductive systems?
Do you have different kinds of waste going each way? Or is it just...
Yeah, most of the time water is going to go through the flame cell system. The ammonia, I believe, can go either into the intestinal ceca or the through the tegument. You can take some ammonia waste with you in the excretory or in the flame cell system, but I don't think that's a primary way of the removal, waste removal. All right, reproductive system. We're going to talk about the male system, and then we'll move to the female system. I'm going to say the male system is pretty simple. All right, simple order. We got the testes. They're going to produce the sperm. So we've got our testes down here. They're going to produce the sperm. Sperm are going to travel through the vas efferens. One tubule from each testes goes, and then they join up. Both vas efferens join up to the vas deferens. So if you've had anatomy, it's the exact same human anatomy. Vas efferens, they join up, vas deferens. Then leads to the seminal vesicle. Your sperm are going to hang out, wait until reproduction. You do have a prostate gland that surrounds the tubule. And the tubule goes into the serous, which is housed in the serous sac when it's retracted. All right, so the serous is protrusible. It's our copulatory organ in our worms. All right, so you've got the duct, you've got your vas deferens going through, gets to your seminal vesicle, and then you have your ejaculatory duct through that serous. All right, so the serous is going to come out of the genital pore, the shared genital pore. All right, and it's going to deposit sperm into the female. All right, and the trematodes, this serous is going to transfer the sperm right into the other genital pore, into the other worm's genital pore, and deposit sperm, and then sperm's going to swim up through the uterus to get to the seminal vesicle if there is one, possibly the lawyer's canal if there is one, or just kind of goes right to the oocyte and fertilizes ova that are coming through. All right, the serous sac can be visible. Sometimes you see the outline even when the serous is extended out. Oftentimes when the serous gets reverted, you can actually see the outline of that serous. And we have several worms where you can make out the serous sac itself. All right. Questions on the male system? So terminology, well, really the only difference here is our serous. It's our copulatory organ. The difference to human anatomy. All right, female reproductive system's a bit more complicated. All right, so we start off with the ovary. note in the male system, I said testes plural, testis is singular, right? Because you typically have two testes, two or more testes. Our females, we typically have a single ovary. That ovary is going to produce the ovum. That ovum travels down the oviduct to get to the ootype. Along the way, it's going to pass the branch that is the vitelline duct. And the vitelline duct goes to the vitellaria, or the vitelline glands. All right? So the egg travels down the oviduct. It's going to pass the branch for the vitelline duct in the vitellaria. It might also see a seminal receptacle and or a lawyer's canal along the way. Doesn't have to. They're not present in all of our worms. All right, and I'm going to define all of these things. That's in our next slides. All right, so here we've got our ovary. You have our our oviduct here traveling right through here. In this case, we have the seminal receptacle branching off of it. We have the lawyer's canal branching off of it, and then we have our vitelline duct branching off of that, and then we get to our ootype. Now, it's the ootype, and it has a gland that surrounds it called the Malus gland. All right? In our worms itself, it'll be very hard to distinguish between those two, because they're basically, if you can think of the ootype as a ball, and then the fur around that ball is the Malus gland. And that's kind of how you see it. You've got glandular cells around that. All right. Once you leave, so fertilization is going to happen in our ootype, and you have the start of egg formation occurs there, and then that newly formed egg, or the developing egg, enters our uterus, matures as it moves through and passes along, and then it gets to the metroterm. The metroterm is the muscular vagina, or it's a muscular structure that acts like the vagina of the worm, and then it opens up into this genital pore, shared genital pore. So both male and female open up to the outside at the same place. All right. So what are all of these terms? So this is the general flow diagram of the female system. Let's go through what each of these does. So hopefully seminal receptacle is self-explanatory. It's going to store the sperm. It's going to receive the sperm, and it's going to store it. And then sperm will be released as an ovum pass, and they'll join up in the ootype. Lawyer's canal is rather interesting. Lawyer's canal is a blind duct, or possibly it could open at the surface. Its function is a little unknown. It's been postulated that it could have been a vestigial vagina, that you've got a separate genital opening where the eggs come out, and then you have this vagina where the serous can enter and deposit sperm right close to the ootype. All right, maybe that's why it's there. You know, maybe we're seeing the loss of it. And again, its function seems like it could also store sperm. And those species that have a seminal receptacle, they can find sperm in that lawyer's canal. Those that don't, you can find sperm in that lawyer's canal. So could store sperm, doesn't have to. Just kind of a big question mark as to what that is. We have one worm that has this lawyer's canal. It's hard to see in all specimens. So if it's on the word list, take a look at numerous slides. You should find it. You'll be able to see it. The vitellaria, or the vitelline glands. These glands are going to supply the nutrients for the yolk, as well as substances that will later become the eggshell. All right. This is the reason why we have that ectolecythal development. Right? The ovum isn't providing the nutrients for the developing embryo. The vitelline glands are. Their location varies by species. We've seen some where it's located near the periphery, near the outer edges. Some of them extend the entire length of the worm. Some of them are confined to, let's say, a region between the testes and the ventral sucker, as it is in this diagram. Ready? Ootype. This is where the eggs are fertilized. This is where everything kind of starts to come together. Your vitelline follicles, the yolk part of it, your ova, and the sperm. All join up, eggs get fertilized, and then the rest of the compounds from the vitellaria, kind of the globules surround the egg and will start to form the shell. All right. Now, also here is this Malus gland. It's an unknown function, but it's glandular tissue, glandular cells, so it's assumed that these things are going to secrete some compounds. All right. The thought is that it could secrete some adhesives that allows the yolk and the other vitelline follicles to kind of stick together to kind of form a compact eggs, until the shell actually starts to form, kind of holds things together. Or perhaps they're secreting some compound that acts as lubrication. Now, you can see some of our specimens, the eggs are really packed in there. Right? You're really packed in there, they're moving around. Need a little bit of lubrication to kind of help them move through the uterus to get to the metroterm. And speaking of the metroterm, this is our muscular end. All right. Two purposes really. One kind of functions as a vagina. Sears is going to go in there, can kind of help grasp hold until sperm gets transferred. The other function is an ovojector. So it's muscular, it keeps the eggs in the uterus, and then when it gets ready to drop the eggs, it can expel the eggs one at a time. I have seen some of our slides where you see the uterus it has a bunch of eggs all bunched up, and then all of a sudden you start seeing single eggs that look like they're floating. You're in the uterus and they are starting to line up singly because that's how they would come out of the worm. We'll see it pretty clear when we get to nematodes, because the nematodes also have the metroterm. All right, we good? All right, life cycle. So we'll see all of this. These are kind of start reviewing these terms. This is part of, what's the structure, what's the function as part of the lab. All right, life cycle and development. We're going to probably end with this slide today. So life cycle is always indirect. It always requires two hosts, and as its subclass name, the Digenea, indicates, we have two generations. We're going to have sexual reproduction that occurs and asexual reproduction. Sexual reproduction is going to occur in the definitive host. That sexual reproduction is going to be meiosis. Meiosis is going to happen, and then you're going to have fertilization to produce a zygote. So having and then doubling to get back to that 2N. Not everything's 2N though, so kind of warn you about that. Asexual reproduction occurs in our first intermediate host, so it's going to be in that mollusk. All right, and this isn't just asexual reproduction in the terms of reproducing without a mate. This is polyembryony, cloning, basically. All right, this doesn't involve self-fertilization at all. This is, you have one individual and it's going to basically clone itself repeatedly to produce our next larval stage. Now this is hugely important because that next larval stage that we're talking about is this guy, this cercaria. It's going to be this cercaria that is also ultimately the infective stage. So if we go from the vertebrate and the snail, getting back from the snail to the vertebrate needs a lot of individuals to make it likely that we get there. Probability of one getting there is going to be super small. Right? Might be 1 in a million. But if you produce 1 million, asexual stages, then the estimate is at least one of them will make it. So our asexual reproduction is serving to really increase the chances that at least one of our larval stages gets to that next host so that this adult can be successfully reproduced. Now, there's going to be much variation in our life stage sequence, all right? And what I'm talking about is that sequence that occurs inside the snail, all right? So typically, in our generic life cycle or generalized life cycle, we have the adult producing the egg. So the egg hatches, release a miracidium. That miracidium produces a sporocyst. The sporocyst produces a rhedia. Rhedia produces cercaria. Cercaria develops a metacercaria. That metacercaria becomes the adult. That's our generalized life cycle. But with all of the variation, there are some stages that can be removed. Sporocysts are always going to happen. You always go from miracidium to sporocyst. But sometimes these sporocysts are going to just start producing cercaria immediately and skip out this whole radial step. Sometimes these sporocysts are going to reproduce and produce other sporocysts. So you have the mother sporocyst that develops from the miracidium. Those mother sporocysts produce daughter sporocysts, and all of those daughter sporocysts then produce the cercaria. The mother sporocysts, they are programmed. You're either producing a sporocyst or you're producing the cercaria, and it's very precise. It goes mother sporocyst to daughter sporocyst, then the cercaria. Sometimes the sporocyst, that mother sporocyst, produces rhedia. And then those rhedia can, just like the sporocyst, produce the cercaria directly, or those rhedia are mother rhedia that produce daughter rhedia, and then the daughter rhedia produces cercaria. And in our last variation, you've got mother sporocyst producing daughter sporocyst, that produce mother rhedia, that produce daughter rhedia, that produce our cercaria. So now you're looking at multiple asexual reproductive steps that exponentially increases the number of cercaria that gets produced by that one individual. All right? So what we will do is pick up with this and talk about all of the different stages, life cycle stages. To find them, have some images of them, and that's kind of in preparation to our platyhelminth diversity. All right, let's go back and review it. If you have any questions over this material that we presented today, please bring it with you on Friday and we'll answer the questions first. All right, see some of you in lab.